Good day, this is Professor Will, CBMD PhD, and today's Wednesday, April 20th, 2022. It is 5:54 and 45 seconds, a.m. Eastern Standard Time, and today's research, the human genome. So let's hop to it here. Researchers generate the first complete, gapless sequence of a human genome. Date March 31st, 2022. Source: National Institutes of Health. The scientists have published the first complete, gapless sequence of a human genome, 2 decades after the Human Genome Project produced the first draft human genome sequence. According to researchers, having a complete, gap-free sequence of the roughly 3 billion bases or letters in our DNA is critical for understanding the full spectrum of human genomic variation and for understanding the genetic contributions to certain diseases. Analysis of the complete genome sequence will significantly add to our knowledge of chromosomes, including more accurate maps for live chromosome arms, which opens new lines of research. And this helps answer basic biology questions about how chromosomes properly segregate and divide. The T2T Consortium used a new complete genome sequence as a reference to discover more than 2 million additional variants in the human genome. And these studies provide more accurate information about the genomic variants within 622 medically relevant genes. And generating a truly complete human genome sequence represents an individual scientific achievement, providing the first comprehensive view of our DNA blueprint. And this foundational information will strengthen the many ongoing efforts to understand all the functional nuances of the human genome, which in turn will empower genetic studies of human disease. The now complete human genome sequence will be particularly valuable for studies that aim to establish comprehensive views of human genomic variation or how people's DNA differs. And such insights are vital for understanding the genetic contributions to certain diseases and for using genome sequence as a routine part of clinical care in the future. Many research groups have already started using a pre-release version of the complete human genome sequence for their research. The full sequencing builds upon the work of the Human Genome Project, which mapped about 92% of the genome, and the research undertaken since then. And thousands of researchers have developed better laboratory tools, computational methods, and strategic approaches to decipher the complex sequence. The 6 papers encompassing the completed sequence appears in Science, along with companion papers and several other journals. That last 8% includes numerous genes and repetitive DNA and is comparable in size to an entire chromosome. The researchers generated the complete genome sequence using a special cell line that has 2 identical copies of Earth chromosome, unlike most human cells, which carry 2 slightly different copies. The researchers noted that most of the duly headed DNA sequence were near the repetitive telomeres, long trailing ends of each chromosome, and centromeres, dense middle sections of each chromosome. Much more, ever since we had the first draft human genome sequence, determining the exact sequence of complex genomic regions has been challenging. The complete blueprint is going to revolutionize the way we think about human genomic variation, disease, and evolution. The cost of sequencing a human genome using short-read technologies, which provide several hundred bases of DNA sequence at a time, is only a few $100s, having fallen significantly since the end of the Human Genome Project. However, using these short-read methods alone still leaves some gaps in assembled genome sequences. The massive drop in DNA sequencing cost comes hand in hand with increased investments and new DNA sequencing technologies to generate longer DNA sequence reads without compromising the accuracy. Over the past decade, 2 new DNA sequencing technologies emerged that produce much longer sequence reads. The Oxford Nanopore DNA sequencing method can read up to 1 million DNA letters in a single read with modest accuracy, while the PacBio Hi-Fi DNA sequencing method can read about 20,000 letters with nearly perfect accuracy. By using long-read methods, researchers have made breakthroughs in our understanding of the most difficult, repeat-rich parts of the human genome. And this complete human genome sequence has already provided new insight into genome biology, and we look forward to the next decade of discoveries about these newly revealed regions. Furthermore, the finishing effort, sequencing a person's entire genome, should get less expensive and more straightforward in the coming years. In the future, when someone has their genome sequenced, we will be able to identify all the variants in their DNA and use that information to better guide their healthcare. And truly finishing the human genome sequence was like putting on a new pair of glasses. Now that we can clearly see everything, we are one step closer to understanding what it all means. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.